So, first of all, it was already mentioned that I was part of this National Alzheimer's Coordinating Center. Um, Please take a look at the website, and you can learn more about what that is and what we do every day to help out with that. We do coordinate the 36 Alzheimer's disease research centers around the country, as you can see from the map and so on, and have been doing this a long time. Well, today, to get right into it, we're gonna walk through a little bit of a brief history that I've put together. Also show you some pathology pictures, some slides, and talk about a little bit about the changes in diagnostic criteria and how that affects how we talk about Alzheimer's disease and what is symptoms versus what is the actual underlying disease. We're going to talk a little bit about the disease continuum and where different interventions might be made to observe potential effects. I'm going to show you a tiny bit about genetics, just in terms of a couple of indicators for familial Alzheimer's disease versus what we call late onset Alzheimer's disease. And I'm going to talk about something that's just really come to the fore in the last couple of years. That's multiple etiologies. That is, usually for most people, there's more than one underlying disease that is causing the cognitive problems or the dementia, if you want. And then to get a little bit more modern, we talk about the biomarkers that we have, actual disease biomarkers, not just sort of things that are associated, but things that actually denote that the disease itself is present. And then right at the very end, I'll give you a little bit of what we finally have as the new disease modifying treatments. So I guess we might as well get started, and you can ask me questions later on. Here's Dr. Alzheimer about 1906, and he had a patient who had followed for about 4.5 years. And her name was August Dieter, actually, and she had kind of an unusual paranoid kind of symptom complex and so on, and he followed her for a long time. It was kind of unusual. When she did die, he did an autopsy and examined her brain, and he found what he called these peculiar amyloid plaques and neurofibrillary tangles. This is August here, and during that time, because she was only 51 years old, they began calling this dementia precox, or actually just early onset dementia, which they thought at the time was different from what they called senile dementia or dementia among the older, as people got into their 80s or 70s or wherever. Later, she was discovered, much later than this, it was discovered that she had this particular mutation for familial Alzheimer's disease. She had a little bit different presentation in terms of symptoms, in that she had more paranoia and agitation than memory problems, if you will. So I'll see if I can walk you through a bit of this. So here's Dr. Alzheimer's and his peculiar deposits in 1906. He left Tübingen. He presented this in Tübingen, Germany in 1906. I was there in 2006 for the 100th anniversary celebration of that particular lecture. In 1910, he went to Munich, I believe, And that's where Dr. Kraepelin, one of the original neurologists, I guess, viewed Alzheimer's disease as an organic cause, kind of as opposed to what Freud might have called another psychiatric cause. So, but this was all considered to be a pre-senile dementia and kind of a very rare sort of condition, all the way from 1910 till about 1976 or so. And in 1967, Tomlinson and Blessed and Roth looked at a couple hundred brains of older individuals who had different sorts of dementia. This was in the UK, and they found out that, well, the same sort of deposits were in those older brains as Alzheimer had found in the early ones. In about 1976, Bob Katzman in New York at the time looked at a lot of these senile dementia cases, as they were called at the time, clinically and neuropathologically, and he said, well, wait a minute, these look almost the same, and so maybe this is just a continuum of the disease about the same disease as we expand the age group. About 1976, also, Peter Davies said, well, gosh, against this backdrop of what we're doing to characterize and treat Parkinson's disease with L-DOPA and similar analogs, maybe there's something that we can treat the dementia people with to replace neurotransmitters, and maybe that will solve the problem of dementia. And actually, that's the basis for the treatments that we have today, except for these brand new disease-modifying treatments. So things like Aricept are based on replacing or keeping enough supply of acetylcholine at the synapses of the neurons. Okay, in about 1984, George Glenner and Connie Wong, his laboratory discovered what this actual structure of amyloid was, the beta amyloid that formed those plaques that Alzheimer had seen. And they also noted that in Down syndrome people who are trisomy 21, three copies of chromosome 21, that they also had similar amyloid deposits. In about 1987, just right about the same time, there was a whole flurry of genetic work going on at the time. It was discovered that the amyloid precursor protein gene was on chromosome 21. And so that encouraged a lot more work to discover a familial Alzheimer's disease gene. So looking in big families where there was multiple affected cases, trying to do segregation analysis and those kinds of techniques at the time to determine what genes were involved. In about 1990 to 95, two particular genes, presenilin 1 and 2, were discovered that were actually autosomal dominant familial Alzheimer's disease genes. So, let's see what happened. Something happened here. There we go. So at any rate, presenilin 2 was actually discovered here at the University of Washington. Tom Bird, Jerry Schellenberg, Ellen Weissman, George Martin were primary leaders in that particular effort. In 1993, Alan Roses and his team, Warren Strittmatter, Beth Corder, and so on, discovered apolipoprotein E, which had been thought to be a cardiac-related gene, but they noted that people who had a particular allele, the epsilon 4 allele, tended to have increased risk for Alzheimer's disease. Kind of in parallel with that, also Heiko Brock and his lab, Brock and Brock, discovered that, or looked more at the tau protein, the neurofibrillary tangles that Alzheimer observed, and developed a means of staging the spread of neurofibrillary tangles from deep inside the brain, the medial temporal lobe, up to the cortex. So the 6 Brock stages were coined in that evidence. And then more characterization of beta amyloid, because that was the particular active ingredient of the amyloid that was causing the plaques, became a drug target and began to influence a lot of studies to see if that could be eliminated by applying different sorts of antibodies. And then the Alzheimer's clinical symptoms were also looked at in more detail so that we could look at that more closely. Okay. In about 2001, we started looking more at, can we recognize these symptoms earlier? Because we thought if we could find them earlier, we might be able to treat them earlier and perhaps have better success. So Ron Peterson and others coined the term mild cognitive impairment. So this was an impairment that was noticeable, but didn't necessarily severely affect actual social occupational functioning. In about 2003, 2005, Bill Klunk and Chet Mathis from Pittsburgh developed a compound that was based on the pathology stain called Congo red. It was a radioactive ligand that would bind to the amyloid in the brains of people that had amyloid. And so they called it Pittsburgh Compound B. It was labeled with carbon 11, and so would light up those amyloid deposits in the brains of living people. And that was a really major breakthrough. It had never been able to do that before, and couldn't really visualize or determine these deposits until neuropathology or post-mortem examination. So that's led to even more other ligands that are related to look at both amyloid and the tau that makes the neurofibrillary tangles. There was also improvements in MRIs, so that more things could be looked at in terms of vascular disease and so on than just big strokes and that sort of thing. Also, Kai Bleno and others were looking closely at cerebrospinal fluid measures of amyloid and tau to see if they could use those as biomarkers to identify people who had the disease. A little bit later than that, there was also new ligands for imaging tau deposits, the tangles, and visualization of that. So people started to characterize the disease in a research framework as amyloid, tau, and neurodegeneration, or what they could see how atrophy was occurring, the neurodegeneration. Drug trials started after that, looking for anti-amyloid agents, primarily antibodies against the beta amyloid that accumulated. And there was evidence that was developed in the DIAN study, dominantly inherited Alzheimer network. So those familial Alzheimer's disease cases I talked about, and this study was headed by John Morris and Randy Bateman at WashU. And from looking at those families where people had the autosomal dominant familial Alzheimer's disease, they were able to estimate that the actual disease process, or the extra formation of the plaques primarily, may start 10 to 25 years prior to any cognitive symptoms. Okay, let's see. And then I have a little trouble with my screen here because I can't read the rest of the things here. So Alzheimer's disease frequently occurs not just by itself, which at the time we always tried to limit it and say, well, is it Alzheimer's disease or is it something else? But actually, Alzheimer's disease, underlying disease, also occurs with vascular disease, Lewy body disease, and a variety of other things. Another big breakthrough came just in the last couple of years, and actually the FDA approved of a blood-based biomarker for amyloid and tau, where a blood test could be done to show that whether or not, with some degree of accuracy anyway, that you had that process going on. Need to be verified probably with a PET scan, but still pretty highly sensitive and specific against a PET scan. And right now, or just in this last couple of years, the first disease-modifying treatments were developed, lecanemab and donanemab, that get rid of amyloid plaques and also help a little bit anyway with cognition. So that's the kind of my long brief history here, but that should open up a lot of other things. There are some pictures here. Here's a lateral view of a brain of a patient with advanced Alzheimer's disease. You can see, I don't know if you can see the deep grooves here, the sulci, how it's kind of shows the degeneration, especially up here in the cortex, not so much back here in the occipital lobe, or not so much down here in the cerebellum, but a lot of degeneration. Here's a coronal section, just so like a cut across there of a section. This is a pretty advanced case. You can see a lot of degeneration going around here, big ventricles and so on, not much in hippocampal areas and so on. A lot of degeneration. Here's a picture of a normal cerebral cortex, and here's a picture of a cerebral cortex that has a lot of amyloid plaques, these little dark spots. Okay, this is a higher magnification over here. This is what would be called an amyloid plaque or a neuritic plaque, basically, a more mature form of amyloid. And this other thing right here is a neurofibrillary tangle, which is hyperphosphorylated tau protein. This shows that these circular sort of structures are blood vessels, cross-sectional blood vessels on a pathology slide, and the yellow part around here, around each one of these blood vessels, is actually the amyloid is up here in this picture, and it forms on the inside of those vessels and sometimes causes hemorrhages or causes less circulation in these very small vessels. So this is called cerebral amyloid angiopathy, or CAA. Okay, here's another picture of these and kind of the characteristic shapes of neurofibrillary tangles. They have this flame sort of shape. So the neurofibrillary tangles and the amyloid plaques were the characteristics that Alzheimer noted. Here's something else that you may be interested in: alpha-synuclein deposits. These are called Lewy bodies. These are two different stains, immunohistochemical stain and ubiquitin stains, to identify these Lewy bodies, which are deposits of alpha-synuclein. And why they're important is because in probably about 40% of the cases that have Alzheimer's disease, they may also have Lewy bodies somewhere in their brain, either in the cortex or and close by to the others, or maybe down in the amygdala or some other areas. But they coexist fairly frequently, and you've probably heard of dementia with Lewy bodies, as well as Parkinson's disease. So there's a lot of overlap in these etiologies. Okay, well, where do most people go if they start to have cognitive symptoms or some sort of other symptoms? Their family brings them into a clinic or does something. Well, most of them are just, of course, first go to primary care. And usually primary care is such a busy place that the primary care docs don't have a lot of time to spend with this, and so they might refer to other specialists, maybe to geriatrics, maybe to psychiatry, maybe to neurology. Maybe they'll go even to a research clinic. A very small number of them might go to the local Alzheimer's disease research center, for instance. But you can see that what gets handled in primary care is really a lot, and sometimes they don't have the time to really nail down what's actually going on. And early on in the development of the disease, probably it's just left to well, just take care of them, take plenty of time to make sure that they're kept safe and so on, and we'll follow it in another year or so and see what happens. Okay, so how do we start nailing down this a little bit so that what one area was calling Alzheimer's disease was sort of the same, at least, as what other people were calling as Alzheimer's disease, so we could study it. We had relatively standard diagnosis. And in about 1984, there were the NACC ADRDA criteria, Guy McCann and a big working group. And they created these criteria for what they called probable and possible Alzheimer's disease. So they had to have serious cognitive deficits, and it was only considered definite Alzheimer's disease when there was a post-mortem exam. These criteria are similar to what some of you might be familiar with as the Diagnostic and Statistical Manuals, the DSM-3 and 4, and now 5 is out. The pathological diagnosis of Alzheimer's disease required that they have symptomatic dementia in addition to just those neurofibrillary tangles and the neuritic plaques of amyloid that Alzheimer noted. And there was a couple pathologic criteria, one by Kachaturian and the CIRAD criteria, and also even the NIA Reagan criteria for the pathologic diagnosis in 1997 required that the person also have dementia symptoms in addition to that in order to be called Alzheimer's disease. However, in 2011, the pathologic criteria were based only on the biologic occurrence of amyloid, tau, and degeneration, or neuritic plaques. So in 2012, came some new clinical criteria that included cerebrospinal fluid and PET, or positron emission tomography, biomarkers for amyloid and for tau, to be considered in combination with what had been described as the clinical syndrome. So dementia or severe functional deficits in social and occupational functioning due to cognitive problems, with the support of these biomarkers, or these mild cognitive impairment that I mentioned a while ago, also with biomarker support. So maybe they have positive amyloid or tau. And then it left the door open for these preclinical Alzheimer's disease biomarkers to occur to establish, well, maybe this is consistent with an asymptomatic presentation of the disease. Okay. In about 2018, these criteria were updated again to reflect more specifically the Alzheimer's, tau, and neurodegeneration framework, getting closer now to a biological definition of the disease. And those still had those similar criteria. And then just in the last year, these have been updated again to be more specifically based on pathology or biology, if you will. And this is the reference for those revised criteria, which allow for the diagnosis to be made in the absence of any symptoms. So these new criteria in living people, the disease is diagnosed by, oops, wait a minute. My screen just, there we go. Okay, the symptoms are a result of the disease process. Oops, having trouble here, sorry. Symptoms are a result of the disease process. They're not necessary to diagnose Alzheimer's disease. Unimpaired individuals with abnormal biomarker testing are at risk to develop cognitive and dementia symptoms due to Alzheimer's disease. They're not at risk for the disease because they already have it. Clinical syndromes commonly seen with Alzheimer's disease may also be caused by other disorders that are other than Alzheimer's disease, and therefore clinical presentation alone is not diagnostic of Alzheimer's disease. That is to say, severe memory problems don't necessarily mean it's Alzheimer's disease. It could be Lewy body disease, it could be other sorts of things, or it could be a combination of a variety of things, vascular disease as well. And even the same Alzheimer's disease biology may result in slightly different presentation of symptoms or phenotypic presentation. Okay, so if we can get this straight as far as nomenclature goes, just in the last couple of years, there's been a change in how we describe the condition. So Alzheimer's and dementia are often used interchangeably, but that's under the newest criteria really not deemed to be correct. Dementia is a set of symptoms, cognitive symptoms, And Alzheimer's disease is the underlying pathologic process or brain disease that generally begins without symptoms and maybe, say, 10 to 20 years before any symptoms. Dementia is a progressive loss of abilities, serious enough to interfere with daily life and something that's different from what the usual functions for most of their life was. Alzheimer's disease is the most common contributing etiology, but there's other neurodegenerative and vascular conditions that usually coexist. And a lot of the earlier criteria didn't allow for that. It was kind of a diagnosis of exclusion. So we break this down into two axes, if you will, a clinical symptom axis and an etiologic disease axis. So the symptom axis goes from normal to mild cognitive impairment to dementia, and it's a continuum of cognitive change with age, but also being pushed by underlying pathology. And it may get worse and worse as it progresses. It can be affected by what we call cognitive reserve, or how resilient the person may be to that pathology that's developing. And that might be related to their general health, their education, their social functioning, a variety of things. The disease axis, however, may have Alzheimer's disease, vascular brain injury like stroke, small vessel disease, CAA that I talked about, Lewy body disease, frontotemporal lobar degeneration, which includes all these other behavioral frontotemporal dementia, primary progressive aphasia, and so on, even ALS, chronic traumatic encephalopathy, and repeated head injury, another cause of disease, also HIV, prion disease, mad cow disease, and Huntington's disease, and so on. But all of this forms this idea of multiple etiology dementia, multiple etiologies causing symptoms. Okay. So how do we know clinically or observably if cognition is declining? Well, we might ask the patient or their spouse or some close contact. A lot of people, as they age, just get to be like worried, well, almost everybody forgets something. And so there's going to be some of that. But when it gets a little bit more severe, when it's really affecting how people function day to day, maybe that's a problem. So we ask about daily living functions. How do people do ordinary kinds of things? Are they able to prepare a meal that is something like they usually would have done in the past? Do they, they always ask, well, do they balance a checkbook? Well, nobody has checkbooks anymore, but something similar to that, I suppose. You can also send them to a neuropsychologist and do some cognitive tests. You've probably heard, the president talking about taking his MOCA test and doing wonderfully on it, and asking a little bit about kind of a blunt instrument, asking about memory, asking about attention, executive function, verbal learning, that sort of thing, to see whether the scores people obtain fall outside what might be considered a range of normal, to give the clinician an idea that maybe there's something going on. Also, sometimes people have trouble with language, aphasia of different sorts, particularly word finding difficulties and so on. Maybe they do some unusual things that you wouldn't normally sanction in a public setting. They have some behavioral comportment issues. Okay, so maybe then we'll send them to a neurologist and get a neurology exam and see whether there's other sorts of things going on. See if they have a movement disorder. See if they have some other functional disorder that can be determined from a neurologic exam? Are these different from their usual and customary functioning? If somebody's been, a little bit affected through their whole life, we wouldn't make the same sort of inference as we would as if this is something that's developed over the last several years, for instance. The main big question that separates, say, MCI from dementia is whether these deficits in cognition are affecting social and occupational functioning. Are they able to just do the normal things with other people or in an occupational setting that they could do before the onset of the problems? Okay, here's a little bit of a picture of how we expect this clinical dementia syndrome axis to take off. So the trajectory, there's always some, you can expect, well, there's some decline with aging, but when it's maybe more pathologic, it declines a little bit more steeply and passes through this preclinical area where there's just a little bit of deficit or dysfunction, and then into MCI, and then down here into dementia. And it's important here to think about this in another sort of a way. So I've drawn another picture here, a disease continuum from no disease here on the left end to probably terminal disease over here on the right. And where we started out to describe most of this disease was right over here on the right-hand side. What did this disease look like? Well, it was pretty severe. It had all these findings in the brain and so on. And there was all these symptoms. Even when we went a little bit earlier to look at MCI due to Alzheimer's disease, maybe it was detectable a few years in advance. Well, now with the newer methods, the newer biomarkers that we're able to mobilize, we can detect disease, this underlying disease, in the asymptomatic phase before symptoms began. Okay, so that's this secondary prevention window, and that's the window that we would probably, or the window that we now intend to treat with the disease modifying therapy. Okay, treating with Aricept and the cholinergic cholinesterase inhibitors and so on here in dementia helped a little bit, provided a little bit more of neurotransmitters to already dying neurons, so that it helped a little bit. But the idea with the disease modifying treatments is to affect the amyloid deposition in this area to keep symptoms from developing or slow the process of symptom development. Okay. Ultimately, we'd like to do something with primary prevention, but nobody's quite into that area yet because it's just much harder to do. What could we give people in advance of any neuropathologic evidence of disease to prevent it, like a vaccine? Nobody's really working in that area yet. And likewise, nobody's been able to do anything with severe dementia and treating them in order to recover and bring them back to a preclinical state. Okay, there's lots of things that could potentially be interacting and modifying to cause all of this whole system to happen. So I'm just sort of outlined them here. There could be what we call the internal exposome, or things revolving around what's in your body, or how you're doing with the medical problems, your metabolome, your history, all of that. There's external factors like environmental toxins, occupational exposures, your ability to do things, your your education, your diet, your lifestyle, all sorts of things, and the external things. And then those all may influence manifestations of the disease, as we've talked about already. Okay, so we've been talking about amyloid, and everybody has an amyloid precursor protein gene, or actually probably more. We look at the APP gene that it produces this amyloid protein normally, but the problem is that sometimes it's not metabolized normally. So when the APP gene mutations or other defects in the metabolism are active, then it forms this A beta peptide, we call it, or a chunk, amino acids 1 to 40 or 1 to 42, and it's produced in a pathogenic way and causes that pathogenic amyloid that forms the plaques. These A beta bits bind together and form dimers and trimers and oligomers, eventually protofibrils and then fibrils that turn into plaques, which are insoluble misfolded proteins that I showed you on those pictures. These compromise vulnerable tissues like neurons, so cause degeneration of neurons. Okay, here's a cartoon that Jerry Schellenberg gave me one time. This is in the middle, this is like what the amyloid precursor protein itself looks like as it spans the cell wall. Okay, so here's inside the cell and here's outside the cell. When it's cleaved normally, something by like this enzyme called alpha secretase, it's cleaved right here, and this part is just degraded normally, and no A beta peptide is formed. However, when it's cleaved in the wrong place by gamma secretase, which might be presenilin 1, and by beta secretase, another enzyme, it forms that A beta chunk that eventually forms fibrils and so on and develops into an amyloid plaque that is sensitive to neurons. Okay, so I mentioned about the autosomal dominant form of Alzheimer's disease. It's actually quite rare. There's maybe about 1% of the cases observed, maybe even less than that, are this autosomal dominant form. And so early on in the study of this kind of disease, the autosomal dominant form, it was hoped that we could learn something also about the more common form and draw some of those findings into helping us solve the problem of the more common form, the non-autosomal dominant. Okay, and that's what I mentioned about Alan Rose's work with apolipoprotein E. And everybody, like amyloid precursor protein, everybody has apolipoprotein E, but they have pairs of these 3 different alleles or different forms of the protein. The E2 form is a little bit protective against Alzheimer's disease. The E3 form is the most common form, and most people are essentially E3 pairs. The E4 allele is the one that's more toxic as far as Alzheimer's disease goes. So you can have pairs of E2, E3, E2, E4, E3, E4, 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 E3, E3, E2, E2, so on. People who have one copy of E4 increase their odds of developing symptomatic Alzheimer's disease by about 3 times. People who have both copies of E4, maybe about 8 times. Okay, so good to remember about that. But it's not like a familial autosomal dominant gene. It just increases the risk. So there's people who have these various forms who do not get symptomatic Alzheimer's disease. Okay, and it works the same way. People who have the other familial form, they also have ApoE, and so it affects their age of onset as well, and does in late onset Alzheimer's disease. A whole lot of genome-wide association studies have been done on very large samples, and most of the studies have shown, significant results based on doing all of these tests, all these various genes or SNPs across all 23 chromosomes, and kind of accounting for chance about whether these are associated or not with the presentation of Alzheimer's disease. So there's some that stick out a little bit more, TREM2 and PICOM and BIN1, but really ApoE is really, this has got a break in it in here because it's way up here somewhere. ApoE is way off the map. Okay, so anyway, we'll move along here. Alzheimer's disease is that A, beta, and tau that we talked about, getting to the clinical spectrum of cognitive, normal, MCI, and dementia. Then I talked about the alpha-synuclein, or the Lewy bodies that kind of go along with Parkinsonism, Parkinson's disease. There's something called TDP43, another protein that misfolds and sometimes creates hippocampal sclerosis, or this rather new clinical characterization of limbic-associated TDP43 encephalopathy, or late. We almost had 2 T's in that, so it became latte, but then they just changed it. So I thought that would be too much for people to handle. So anyway, also vascular disease, very common, and influences this clinical spectrum quite a bit. And then there's a bunch of other things that are less often seen. Like I mentioned, chronic traumatic encephalopathy, repetitive head injuries, a variety of other sorts of diseases and conditions go into this package. So this is this general idea of multiple etiology dementia. And here's the Alzheimer part. There's also another set of these tauopathies that generate their own impact against the clinical spectrum. Okay, so thinking about those multiple etiologies again, pure Alzheimer's disease is uncommon, and this is a study that was done actually by Julie Schneider at Rush, and well, let's see, looking at autopsy data and by age, and the breakdown of kinds of pathology found in people at different ages. So even at about 90, there's a mix of different pathologies pretty commonly. As I mentioned these before, Blessed Tomlinson and Roth, in their early study in the late 60s, found the same kinds of things that Alzheimer had found, but in an older age group. Okay, and locally here, Debbie Swung analyzed a bunch of autopsy data, neuropathology data from the ADPR ACT study that's done here at Kaiser. And looked at the characterization of combinations of different kinds of pathology. So start up here, here's the Alzheimer's alone, that only showed up in about 14%. If you add Alzheimer's and the DLB or the alpha-synuclein, that's another 22% had that mix. Another 12% had Alzheimer's and vascular. 13% had a combination of those three things. Okay, some had purely alpha-synuclein or purely DLB. Some had DLB plus vascular. Some had only vascular. And then these other hippocampal sclerosis or the late disease that I described, combinations of that. And then a couple of these tauopathies, PSP, and some other sorts of things fill in there. And then, unfortunately, in this autopsy series, there was only about 1% that were completely normal. So that's probably partly due to the selection within the study itself into the autopsy. Okay, so we have biomarkers trying to observe these biologic processes in living people. So we have imaging as I mentioned, Klunk and Mathis, looking for amyloid, PET, positron emission tomography. Also, there's tau PET, looking for tau in the same way. There's MRIs that look for neurodegeneration, for vascular disease, stroke, and here's the different sequences of T1, a flare, or DTI, and a whole raft of other sequences. There's also an FDG PET to look at metabolism differences. Glucose metabolism differences, often useful in discriminating between Alzheimer's disease and frontotemporal lobar degeneration. And the view of these things is where the low metabolism, or I have blue is bad here, so rather than being lit up as red or yellow, it's usually the blue parts that are low. And there you can do a DAT scan, they call it, for Lewy body disease, and you get a particular indicator. Cerebral spinal fluid measures that I mentioned, looking for A beta, oops, back up. Looking for A beta, tau, alpha-synuclein. It's getting better to look for that. TDP-43, neurofilament light, a measure of sort of non-specific degeneration, and then and maybe some other proteins as well. And this other huge advance that's happened within the last couple of years, looking at blood or plasma biomarkers for both A beta and phosphorylated tau 181 or 217, also the NFL or the glial fibrillary acidic protein, another measure of degeneration. So how do we know about whether somebody has this biological disease? Well, we can do a PET scan for amyloid and tau. We can do a lumbar puncture and measure amyloid and tau in the CSF. We can do blood-based assays now. And one particular one, this Fujira Bio p-tau217 amyloid beta ratio, which was recently approved by the FDA, is kind of a cornerstone of the new criteria to be like a core 1 level biomarker for Alzheimer's disease. We also do MRIs to look for cerebrovascular disease primarily, or some general atrophy, or maybe some other reasons why the symptoms might be occurring. And so because of the advances in MRIs, we can also do calculated volumes of, like maybe 80 or 90 different structures within the brain. So we can look for cortical thinning, we can look for just different volumes of everything that are calculated automatically by programs like FreeSurfer. We can also look at small vessel disease and infarcts, even large vessel disease. And as I mentioned, those non-specific measures of neurodegeneration, if it's not as evident from the overall atrophy in the MRI, we'd also get measures of neurodegeneration by looking at neurofilament light or GFAP. Can look for Lewy bodies and TDP-43. They're not great tests at this point. They're getting better. The Lewy body tests are just about ready to go. A synuclein seed assay tests, and TDP-43 is a little further off. So, but the good thing about these is these may be positive even if there are no symptoms present. So potentially it opens up that window, and it also sets up another deeper way to look for risk factors, because you'd want to look at factors that occurred before the onset of that positivity of the biomarker, if they were really increasing the risk of onset of the disease. Okay, so again, limitations. Of course, these biomarkers are going to be less sensitive than a neuropathologic exam, but consider that the neuropathologic exam is years later after death and so on, so you wouldn't expect them to be totally matched up. Biomarkers are not available for all those relevant diseases we talked about that might be underlying etiologies. Eventually we'll be able to get those, but we don't have them all at this point. And the proportion of the cognitive deficit in any individual that's attributable to either Alzheimer's disease or some other neuropathologic entity cannot be known with certainty from just looking at the biomarker values. So there's still a lot of work to be done to estimate those contributions and interactions. I wanted to point this out because it kind of brings things together. Cliff Jack from Mayo started drawing these essentially hypothetical in terms of they're all sort of smooth growth curve looking things. But the important thing about here is we look at biomarker abnormality here on the vertical axis and time along the horizontal axis. And you can see as what's developed here, these two lines, two different measures, CSF and amyloid PET, kind of parallel each other, measures of amyloid. And they develop way early on before any symptoms. And it's actually, here we're still at a normal level of cognition, and they're just about maxed out. So these things develop early. We look at tau protein, and it does a similar sort of thing. It starts a little bit later, but these two interact in a way that one kind of drives the other. And so the tau protein also increases dramatically. But again, they're pretty flat by the time MCI occurs, or they're completely maxed out by the time dementia occurs. So here's this green area is the symptomatic area. Here's the neurodegeneration part. This is the atrophy and actually the dying neurons and so on. And even that precedes the symptomatic area. Okay, so this sort of draws the picture, if you want, of how these biologic underpinnings influence the cognitive expression. Okay.
Sorry to interrupt, Dr. Kukull. I wanted to note that it is 1, and I know that we have some questions in the chat. I just didn't know if you would be okay running over a little bit.
I'm okay. Sorry, I just wanted to show a few pictures here. So
okay.
These won't take long, so I just wanted to
Okay.
show, these are PET scans, so tau across the top, and here's amyloid, MRI, and so on. This is a 33-year-old woman, cognitively unimpaired and negative for amyloid, tau, and neurodegeneration, and you can see what those scans look like. Here's a 75-year-old woman, amnestic, multi-domain dementia, okay, amyloid, tau, and neurodegeneration positive. And you can see the tau protein, different views here, but lights up with different colors here. Here's the amyloid, lots of amyloid present. Oops. And you can see degeneration here on their MRI. Here's a person with MCI who's amyloid, tau, and neurodegeneration positive. It's an 88-year-old woman. And we can see the tau light up again, the amyloid, and the neurodegeneration, as you can see around here. And here's a cognitively unimpaired 67-year-old man with amyloid, no tau, and no neurodegeneration. So you can see the amyloid light up here in these different views, but the other ones look relatively normal for tau and so on. So somebody early on in the process. And here's someone with amnestic dementia and no Alzheimer's biology at all. No tau, no amyloid, and no degeneration. So probably has something else, not Alzheimer's disease. And here's an amnestic dementia, or someone who doesn't have amnestic dementia, but has AD biology, a 52-year-old woman with intact memory, intention, and dysexecutive presentation, but with medial temporal lobe sparing and early onset Alzheimer's disease. So lots of amyloid and tau, and the MRI doesn't look so good either, but is unaffected, so asymptomatic at this point. Okay, I think that's all the pictures I have. These are the two new drugs that can be given, and one thing to watch out for with those are amyloid-related imaging abnormalities that cause edema or hemorrhage in the brain that occur maybe in about 20% of the people that take these, and they're given by infusion. Higher rates of ARIA among ApoE4 carriers or homozygotes. Okay, I guess that's about it. So now I'll answer some questions.
Well, thank you so much. Okay, I'll go right into questions since we're running late. The first one I wanted to ask, we had a couple different people ask this. What is the best resource for finding out what we can do proactively to reduce our risk for the disease, whether it's diet, supplements? Yeah.
Okay, well, let me come at it in a couple of ways. I think the old saying, like, what's good for your heart is good for your brain, is probably a reasonable thing to do. So get plenty of exercise and eat a good diet and that sort of thing. That helps to some extent. Also, if you have issues that related to your vascular system, like high blood pressure or something, that's relatively easy and cheap to treat and can really affect that vascular component that might actually kick you over symptomatically and to expression of Alzheimer's disease symptoms. Those kinds of things are probably the most. I don't think there's any specific things. If you wouldn't want to do too much of too much heavy fatty foods, or too much drinking, or too much whatever it might be, because that might affect you. I don't have any great ideas about whether there's whether you should be taking Prevagen or not, but I have no evidence that that works for anything either.
Awesome, thank you. The next one is there an approximate age after which we qualify it as senile dementia and not familial? I can't say the word. Familial.
The familial cases usually usually begin in the 40s, and can span up later than that, but most of them begin in the 40s. And the term senile dementia is kind of an obsolete term at this point, but the ordinary distribution of Alzheimer's disease is usually at the low end in the mid 60s, and then accelerates, doubles about every 5 years beyond that. So mostly 80s and beyond.
All right, thank you. This will probably be the last one, but do we currently have a test to find out if you have any of the symptoms that are not being picked up on the annual health assessment if the patients have 0 symptoms and 0 familial dementia history? Is forgetfulness starting in your 50s sufficient enough to be concerned? That was lengthy.
I'm not a clinician, and I wouldn't advise you specifically on that, but if there are changes in your cognition that are interfering with your ordinary function, then the best thing to do would be to try to see a neurologist, and get a more thorough exam. These newer blood-based biomarkers would be an easy way to determine whether you have the underlying pathology. You have to be a little bit circumspect about just going down and asking for one. But if you had some issues that would prompt it, it would be potentially a good thing to do. Or if there was something of more concern, the neurologist would probably also order a PET scan.
Okay, great. Thank you so much. And thank you everyone for coming. I know we ran over a little bit, so I appreciate everyone staying and asking your great questions. I hope everyone has a great rest of your day, and the slides will be sent out in about an hour. All right. Thank you, everyone. And thank you, Dr. Kukull.
Okay, thanks everybody.
All right. Bye.